Interphase has three parts: gap 1, synthesis, and gap 2. In gap 1, the cell carries out normal functions, increases in size, and organelles increase in number. The cell spends the majority of its time here. It must hit a critical checkpoint before it can go into synthesis to make sure it has enough nutrition, adequate size, and relatively undamaged DNA. These checkpoints are regulated by kinases and cyclins. In synthesis, DNA is duplicated, and by the end of it, the nucleus contains 2 complete DNA sets. In gap 2, the cell continues to have normal functions and additional growth, and it hits another critical checkpoint. In prophase 1, the nuclear membrane breaks down, centrioles move to opposite sides of the cell, spindle fibers form, and chromosomes cross over. In metaphase 1, homologous chromosomes line up along the cell equator randomly as to create genetic diversity. In anaphase 1, homologous chromosomes split and move to opposite sides of the cell. In telophase 1, the nuclear membrane reforms, spindle fibers disassemble, the cell undergoes cytokinesis. The end result is 2 cells with a unique combination of 23 duplicated chromosomes from both parents. After telophase 1, there is a period of time called interkinesis where both gap phases occur, but the synthesis phase doesn't because there is no more need for DNA duplication. During prophase 2, the nuclear membrane breaks down, centrioles move to opposite sides of the cell, and spindle fibers reform. In metaphase 2, spindle fibers align chromosomes along the cell equator. In anaphase 2, sister chromatids are pulled apart from each other and move to the opposite sides of the cell. In telophase 2, the nuclear membranes form around each set of chromosomes at opposite cell ends, spindle fibers break, and cell undergoes cytokinesis. The end result is 4 haploid cells with a combination of chromosomes from the mother and father.